Bismillah ar-Rahman Rahim. We start another question from the March 24 paper 5-2. Now we'll be doing the question 2 in this video. Bismillah ar-Rahman Rahim. Question number 2. Drosophila melanogaster is a small fruit fly that is often used in research on genetics. Why? Because they multiply very rapidly and they're very easy to keep in a lab. You don't need a very big place for them. I mean, you can't have kangaroos for genetic experiments or elephants for genetic experiments. They would occupy a lot of space. Wild fruit flies normally have dark red eyes due to the presence of a brown pigment. So the red eyes due to the presence of a brown pigment homochrome. Wild fruit flies normally have dark red eyes due to the presence of a brown pigment homochrome and a bright red pigment drossopterin. Drossopterin. So dark eyes due to the presence of brown pigment homochrome and a bright red pigment. So the dark red eyes is due to the presence of a brown pigment homochrome and a bright. So the red pigment is drossopterin and the brown pigment is homochrome. Why I'm talking about this? Because this is coming in a later part of the question. So unless you read, I always say in paper 5, most of the answers are in the question and you need to just figure it out. So if you read the question carefully, you can figure it out. So that's why I'll always ask you to read the whole question twice. Figure 2.1 shows an adult female fruit fly and an adult male fly. So here we've got the female and here we've got the male. And you can see this one has the female has a pointed abdomen and the male has a rounded abdomen. So you can see this sort of this round shape and then this little, and it's a little pointed. And you can see this is only 3 millimeter, not very big. Now, what are they going to ask you? A biologist carried out an investigation to determine the roles of two genes that are involved in the determination of eye color in adult fruit flies. Both genes have two alleles. Earlier research by other scientists suggested the synthesis of the brown pigment homochrome depends on the gene big B small b, and the synthesis of the bright red pigment drossopterin depends on the gene big R small r. So the bright red pigment is R small r and the brown pigment is big B small b. And the bright red pigment is big R small r. The biologist obtained the parent fruit fly shown in table 2.1. So all the females were homozygous dominant and homozygous recessive for the second trait. So big B, we have big B and small r, and then you cross them with small b, big R, small r. So you can see what is going to happen. Now they're all going to be heterozygous. No, this is big R. So they will be heterozygous for both characteristics. They'll be heterozygous for both characteristics, the first generation. So when you cross this, the female is this and the male is this. So you cross this, so you get heterozygous for both characteristics. So now we're going to cross this. And then of course, when you cross two heterozygous for both characteristics, the ratio is 9331. So all the offsprings will be this. But of course, when you're reading the question, you've got to figure all this out. You read the question very carefully. Of course, before I did this video, I read this question a number of times to understand the question. So I'm not reading it for the first time right now. Okay. Then what does it say? The biologist carried out the procedure shown in figure 2.2 to cross the parent fruit flies that obtain the first generation of offsprings. And the first generation of offsprings will be heterozygous for both characteristics. Some agar jelly containing nutrients and water was placed in the bottom of several specimen tubes. So the first thing they said was, I'm just repeating myself, some agar jelly containing nutrients and water. Agar jelly containing nutrients and water was placed in the bottom of several specimen tubes. On day 1, 4 adult female with brown eyes and 4 adult male with bright red eyes were placed in each specimen tube. So 4 plus 4. 4 females with brown eyes and 4 males with bright red eyes were placed in the specimen tube. A piece of cotton wool was placed at the top of each specimen tube, right? We've got a cotton wool so that air can enter but they can't fly out. The specimen tubes are left in a warm room for several days. Several days. During this time, the parent fruit flies reproduced and laid fertilized eggs on the agar jelly. The fertilized eggs hatched into larvae that fed on the agar jelly. Right? On day 7, the parent fruit flies were removed from each because you see now they were the larvae, because into larvae that fed on the agar jelly. The larvae still has to become the adult. So on day 7, the parent fruit flies were removed from each specimen tube. By day 18, the first generation of adult fruit flies had developed from the larvae and were ready for crossing to produce the second generation. So we've got as I told you in the last part, we've got the big B, small b, and the big R, small r, and now we're going to cross them. So by day 12, the first generation of adult, this is the first generation, had developed from the larvae and ready for crossing to produce the second generation. So read it very carefully, understand it. Suggest a reason why the biologists removed the parent fruit flies from the specimen tubes on day 7. Now, why did they remove the parent fruit flies? The reason is very simple, very easy, to prevent the parent reproducing and mating with the first generation offsprings. So we want to remove them because now we want the first generation offsprings to mate and result lay eggs, and then we've got to have the larvae and then see what is going to be the next generation going to be like. Okay, now let's read the next. The biologists wanted to cross the first generation of fruit flies with one another to produce the second generation of fruit flies. The first generation of adult fruit flies in the specimen tubes on day 12 were a mixture of females and males and had not yet mated. The biologists crossed the first generation of fruit flies with one another by preparing fresh specimen tubes in which to produce the second generation of fruit flies, using a chemical to anesthetize the first generation of adult fruit flies so that they were temporarily unable to move, separating the adult female from and the adult male. So separating the female and the male, then placing 4 adult female fruit flies and 4 adult male fruit flies into each of the fresh specimen tubes. Now imagine you're doing this in the lab. I always say that's how you get your paper 5 correct. Suggest a method that the biologist could use to separate the anesthetized adult female fruit flies and anesthetized male fruit flies and place 4 of each into a fresh specimen tube. So first, how would you decipher it, and then how would you place them in the specimen? And they're anesthetized, so they're sort of gone off to a little bit, they're unconscious. So you could use a hand lens to first differentiate between the male and the female fruit flies, and then you use a paintbrush you can use a pooter as well, or you can use a forceps as well to pick them up very carefully and put them in the specimen tubes. So in order to get this 1 mark, you had to tell me first how are you going to decide which has a pointed abdomen, which has a rounded abdomen. So you could use a hand lens. You could also use a microscope so that you differentiate between the male and the female. And then you place the fruit flies in the specimen tube. You use a paintbrush or a pooter or a forceps. Then the C part of the question says the biologist expected the cross to result in a phenotypic offspring ratio of 9:3:1. You know that when it's heterozygous for both characteristics, the ratio is always 9:3:1 in the second generation. Table 2.2 shows the results of this cross. So dark red eyes were expected phenotype ratio was 9, the observed number was 691. Brown eyes was ratio was 3, but the number was 260. So this is the number, the observed number. Remember, it's O and E. And the bright red eyes was 225 and the white eyes was 73. So it is a roughly 9331 ratio, and the total was 1248. Now they're giving you the expected phenotype ratio, and this is the observed number. Now read all these, read very carefully, because many of you, as looking at the exam report, many of you talked about the expected phenotypic ratio. Then it says the biologist used the chi-square test to compare the observed and expected results of this cross. State the null hypothesis. There is no difference between the observed and expected results. That is the standard wording that we use for the null hypothesis in chi-square. You can't just say anything else. This is the only sentence that you can write. There is no difference between the observed and expected results. So this is all what you have to write. No difference between the observed and expected results. Then it gives you the formula. The equation of x square is sigma o minus e whole square over e, key to the symbol observed. As it is in the bio syllabus, the formula is always given to you, and you don't have to really memorize the formula like you do it maybe in physics or chemistry. I don't know. I don't know much about physics and chemistry, but I think now it says complete table 2.3 to calculate the value of x square for the results of this cross shown in table 2.2. Give the value of x squared to 4 significant figures. Now, how do we calculate this expected ratio? So 9 out of 16 into 1248. Why? Because 1248 was a total number. So 9 out of 16, because 9 plus 3 plus 3 plus 1 is what? 9 plus 3 plus 3 plus 1 is 16. So 9 out of 16 into 1248. Then 3 out of 16 into 1248, and then 1 out of 16 into 1248. So this is how you would get the expected ratio. Then O minus E whole square. So O minus E, you've got to calculate O minus E whole square over E. Now you have to calculate number one first, the expected value. So that will be 9 out of 16. You did that. Then you O minus E, so that is 11, 26, 9, and 6, and then O minus E whole square, so 11, 11s are 121, 26 into 26 is 676, and then 9 into 9 is 81, and 6, 6 are 36. And then you calculated O minus E, and you get a value of x squared is equal to 3.869. Well, they told you in the question to do 4 significant figures. So you did do 4 significant figures. Now, the biologist compared the calculated value of x squared to the critical values at different probability values shown in table 2.4 using table 2.4. Now, of course, what did you have? You had 1, 2, 3, 4. So it's n minus 1. So 4 minus 1 is 3. So you'd look up in the 3 column and you'd look up 0.05, 5%. Now, this is the critical value. So in 3, why? Because they have number of data is 4. So it's n minus 1. 4 minus 1 is 3. So you look up in the degree of freedom of 3 and the critical value in 0.05, not 0.5. Please remember, it's 0.055 out of 100. So this is the critical value is 7.815. Now your calculated value is 3.869. Now that is less than 7.815. And so the null hypothesis is accepted. If it was more, then it will be rejected. Now it says using table 2.4 and the calculated value of x square on C2, explain whether the null hypothesis is accepted or rejected. I've just told you critical value you have to look up. So the critical value is this one, 7.815, and your value is 3.86. So that is less. So that is why the null hypothesis is accepted. So critical value 7.815, x square value which is 3.869 is less than the critical value, so null hypothesis is accepted. Now coming to the D part of question number 2. More recent research has shown that gene big B small b and gene big R small r code for polypeptides in carrier proteins. These carrier proteins are found in organelle membranes of the pigment cells of the eyes of adult fruit flies. Genes big B small b codes for a polypeptide in the tryptophan carrier. You can see it here. Tryptophan carrier protein. Tryptophan is an amino acid. Gene big R small r codes for a polypeptide in the guanine carrier protein. So this is a guanine carrier protein and this is a tryptophan carrier protein. Figure 2.3 shows how the dark red eye color of wild fruit flies is produced in organelles in pigment cells of the eye. So tryptophan enters, homochrome brown pigment is formed. Guanine enters and drossopterin bright red pigment. So sends a series of catalyzed reactions, enzyme catalyzed reactions, and guanine entering results in drossopterin, and tryptophan, an amino acid which enters into this membrane through this channel protein, results in the brown pigment homochrome. Now read this very carefully, understand it. The biologist decided to analyze all the pigments present in the eyes of the second generation of fruit flies. The biologist started by extracting eye pigments from the adult fruit flies with dark red eyes and the adult fruit flies with white eyes. The biologist then added a small volume of each liquid extract to chromatography paper and separated the pigments present by chromatography. Figure 2.4 shows the chromatography paper at the end of the procedure when viewed using visible light and ultraviolet light. Pigment 1 and pigment 3 were visible only when viewed under ultraviolet light. 1 and 3. Under visible light, pigment 2 was yellow and pigment 4 was bright red. Again, this information must be kept very, very clear in your mind when you're doing the rest of the question. Then we have a figure 2.4 in which you can see the pigment and there's moved different distances. There's a solvent front, there's a chromatography paper, there's a pencil line, and then the position where liquid extract was added from fruit flies with dark red eyes. So the dark red eye seems to have 4 pigments. But the position where liquid extract was added from the fruit flies with white eyes, there seemed to be no pigment. So dark red eyes has 4 pigments. The biologist calculated the RF values for each pigment on the chromatography paper. This biologist uses RF values to confirm the identity of the pigment using a published source. The formula for the calculation is distance moved by pigment, distance from origin to solvent front. When measuring the distance moved by the pigment, the distance to the center of the pigment should be measured. Using figure 2.4, calculate the RF value of pigment 1. Where is pigment 1? Let's look at that. This is pigment 1. So you got to measure the length from here to here, and then you have to measure this solvent from here to here. And please remember it's at the center of it. So this was the center of it, of that pigment. That is a very simple 44 out of 60. And as the answer everybody knows is 0.73. So this was a bit of a maths which they were expecting you to do. So 44 out of 60 and 0.73. Then it says state the conclusions that can be made from the results of the fruit fly by breeding experiment in C and from the chromatography results. Now they want you to look at that and then they want you to comment on the chromatography results. So from the breeding experiment, what did we learn? That the two genes were not linked genes because otherwise you would not get the 9331 ratio. So they were not linked genes. Dominant alleles code for the functional protein. Again, that is a very obvious thing. Fruit flies with dark red eyes only will have the entry of guanine and tryptophan, allowing pigment to be produced. Fruit flies with eyes having the homozygous recessive. Fruit flies with white eyes have the homozygous recessive small b, and small r, small r. So this was the breeding experiment. Then from the chromatogram, what did you guess? Fruit flies with dark red eyes have 4 pigments in their eyes. Let's look at it again. So fruit flies with dark red eyes have 4 pigments in their eyes, and fruit flies with white eyes have no pigment. You can see here white eye, there is no pigment here. Then pigment 4 is drossopteran. Why? Because it told you here in the question. It told you here in the question that pigment 4 was bright red. So pigment 4 is drossopteran, and omochrome, the brown pigment, is insoluble in the chromatography solvent because none of the pigment in the chromatogram are omochrome or the brown pigment. So this conclusions, you have to be very careful in answering this because you have to talk about the breeding experiment, 2 points for the breeding experiment and 2 points for the chromatogram. So in the breeding experiment, you could have given me these points. The breeding experiment told us that, because we're getting the 9331 ratio, so that means they are not linked genes. Otherwise, you won't have got this ratio. Then the dominant allele codes for the functional protein. Fruit flies with dark red eyes, entry of guanine and tryptophan allows the pigments to be produced. And the white eyes must be the homozygous recessive. This is, of course, what we learned from the breeding experiment. Now, um, this would only get you maximum of 3 marks if you give me all the points, any 3 points, and then 1 mark from the other because chromatogram, you had to give me something about the chromatogram as well. So what was the chromatogram telling us? That fruit flies with dark red pigment eyes have 4 pigments. So this was given in the question. It's nothing something which you had to figure it out. It's just reading the question and finding it out from the question. White eyes have no pigments. Then pigment 4 is drossopteran, and brown pigment is insoluble in the chromatography solvent. So that completes question number 2. And I hope this is a little better to you. And please do this question before you look at the video. Please do these questions. Do not just look at the video. Do these questions first yourself or read the questions yourself and then go through the video.